Hello बच्चों, welcome to SPSVERSITY. In the previous session, we discussed about respiration in plants, photosynthesis in higher plants. In this session, we are going to discuss about plant growth and development, which is an important physiological aspect in plants. Okay. So when you come to plant growth and development, so whatever it is, the plant starts its life cycle from zygote. So zygote is the unicellular structure which is formed by fusion of male and female gamete. So zygote on cell division, it will form into embryo. So zygote से embryo form होता है. Embryo is multicellular and which will form, each and every cell will form into tissues and organs. Okay. So from embryo, that is, the cells of embryo will develop into tissues and organs. So what is the difference between growth and development? Okay. So development is sum of the growth and differentiation. So growth is a part of development, where development includes the growth and differentiation. It includes the growth and differentiation. So during the process, it will form a complex body organization. Complex means in which if you look into the plant body, there is root, stem, leaf, branches, flowers, fruits, seeds. These all are formed. So these all are formed from the cells of embryo. In embryo, there is radicle which gives rise to root system. And there is plumule which gives rise to shoot system. शूट सिस्टम comprises of, इसमें शूट सिस्टम में स्टेम, ब्रांचेस, लीव्स, फ्लावर्स, ये पूरा शूट सिस्टम में रहता है. After that they will die. So this is the process of development. पहले हमें देखेंगे what is meant by growth. Growth बोले तो क्या है? Growth is an irreversible change. Irreversible permanent change where there is an increase in size of organs or the parts or even the cells. So during the growth, each and every cell, part or organ will increase in size. और इंक्रीज होने के साथ क्या होता है कि मेटाबॉलिक प्रोसेस भी इंक्रीजेस. सो मेटाबॉलिक बोले तो इसमें एनर्जी का आवश्यकता रहता है. सो इसमें मेटाबॉलिक प्रोसेस के लिए एनर्जी चाहिए. ये एनर्जी के लिए ये बहुत सारे मेटाबॉलिक प्रोसेस उसमें इंक्रीज रहता है. दैट इज by using energy, there is a increase in metabolic process. Whatever it may be. The energy is used for development of shoot system, expansion of leaf, or whatever it may be. The energy is used for various processes which happens in the plant life. So here energy is required for growth. And growth is an irreversible permanent change, that is increase in size. Growth में क्या रहता? Growth में सेल डिवीजन बोले तो सेल से नया सेल्स आना. New cells after this cell division they undergo cell elongation. After that they undergo maturation, which is called cellular differentiation. Elongation means increase in the length. Differentiation means where they attain a specific function. So most of the cells will show mitosis. They divide by mitosis. After formation of new cells, they undergo elongation. The process is called cell elongation. And next is the maturation, the cell differentiation, where they will attain a specific function. So these are the three process happens in the growth. That is cell division, cell elongation, cellular differentiation. Okay. So how to calculate this? Is there any parameter to see how growth has occurred? That is called growth rate. So the increased growth per unit time means you can take either 24 hours or 48 hours or 72 hours. A defined time, the growth increase in the growth is mentioned as growth rate. A growth rate में मैथमेटिकली लिए तो इसमें अर्थमेटिक ग्रोथ रहता और ज्योमेट्रिक, दो टाइप्स रहते, अर्थमेटिक and geometry. Here if you look into the arithmetic, so this is one cell which will divide and form into two cells. So in this only one cell will proceed for the cell division, remaining cell will undergo cell differentiation. The same like whatever the two cells formed from this, again only one cell will proceed for this. The next cell will proceed for cellular differentiation. So like this, whatever the cells are formed, new daughter cells, all will not go for further cell division. Some cells will stop cell division and they go for cell elongation and they go for cell differentiation. So this is arithmetic growth. Geometric growth, which is quite opposite to arithmetic, where whatever new cells are formed, each and every cell will proceed for cell division. here हमें देख सकते हैं एक सेल से दो सेल्स आए, ये दो सेल्स फिर सेल डिवीजन होता है हर एक सेल से और नया दो सेल्स आता है, a new cell से भी और एक दो सेल्स ऐसा it will be each and every cell will divide will proceed for cell division. So इसको क्या बोलते हैं कि geometric growth where all cells will proceed for cell division. Arithmetic growth where only few cells will proceed for cell division. What happens to the remaining cells? They will undergo differentiation, elongation. So here you can see. Generally we can see mitotic cell division. So from this cell, two cells are formed. So this cell will proceed for formation of few cells, that is cell division. But this cell will undergo differentiation. Differentiation means it will attain a specific function. Maturation. So once they are differentiated, they lose the property of the cell division. So once they enter into differentiation, they lose the property of cell division. That is, which is nothing but maturation. The best example you can take the root elongation. So roots meliato, root apex, root elongation, root maturation regenerate. So root apex, meristematic zone, elongation zone or maturation zone. From meristems, root meristems, new cells are formed which will enter into elongation zone. After elongation, they will enter into the maturation zone where they will be attained a function, they will undergo maturation. So in this arithmetic growth, there is only few cells will proceed for cell division. So how to calculate this arithmetic growth? Okay. So here you can take the length of the plant. So length means you can take the length of the root or length of the stem or branch. Okay. So L means length at a particular time. L naught is the time at initial time. R is the rate, T is the time. Okay. So LT means length of the root at a time. L naught means that is length of the root at time 0. 0 means initial. RT, elongation of root per unit time or growth rate. So, by taking length into consideration, we can construct a mathematical expression. That is, LT is equal to L naught plus RT. And if we represent this arithmetic graph in the form of a graph, here if you take time and length, L naught is the initial phase where it is the initial time at 0. As the time proceeds, there is a increase in the growth rate. So there is a increase in the growth rate. So this A by B will give RT value. So this, if you look into the curve, it is a linear curve. So by calculating the length of the root, stem or branch, you can come into a conclusion what is the growth rate in the terms of length. That is, the expression for this is, mathematical expression is LT is equal to L naught plus RT. This is mathematical expression for arithmetic growth. When it comes to geometric growth, here if you look into this, the initial cell which is called mother cell. From this, by mitosis, two daughter cells, the progeny cells are formed. Again from 2 it becomes 4 progeny cells. Again from 4, 8. So every time it becomes double. Why? Because all the cells will proceed for cell division. Starting from mother cell to progeny cells, every cell is in process of cell division. So here there is an increase in the number of cells geometrically. So here if you look into the growth, it is initially slow. पहले स्लो रहता, उसको क्या बोलता है कि लैग फेज. बाद में लैग फेज के बाद में रैपिड इंक्रीज रहता, उसको क्या बोलते हैं कि log phase or exponential phase, followed by again slow down, which is called stationary phase. So इसमें हमें ग्राफ लिए तो एक ग्राफ में एस शेप्ड ग्राफ आता, that is if you take growth and time, पहले स्लो रहता, बाद में स्पीड रहता, बाद में सिग्मॉइड ग्रोथ करवाता. इसको क्या बोलते हैं कि सिग्मॉइड ग्रोथ कर्व, एस शेप्ड रहता. So this is lag phase, this is log phase, exponential phase, and this is again the stationary phase. Okay. So if you look into this mathematical expression of geometric growth, you can take, for example, you can take weight or height or number. So weight, if you take, so W1 is final size at a specific time which we are taking. Okay. W0 is initial stage of the beginning of the period. R is growth rate, T is time, and E is base of natural logarithm. So W1 is equal to W0 E to the power of RT. And if you look into the graph, this is growth and this is time. So here it is the lag phase, here it is the exponential phase, and finally it comes to a constant one, that is stationary phase. So which is a mathematical expression for geometric growth. So geometric growth is where each and every cell will start dividing, will be in the process of cell division. Whereas arithmetic growth, only few cells will proceed for cell division, not all the cells. Okay. Initially, if you take a plant, any plant, initially it will be in geometric growth, where all the cells will divide. But later on it enters into arithmetic growth. Okay. So here if we come to growth rate, there are again two terms in growth: absolute growth and relative growth rate. So what is this absolute and what is this relative growth? Absolute is a direct value, whereas a relative growth rate is a comparative one. Okay. If you look into this, this is a leaf with 5 centimeters square. The initial size A, the leaf size. Later on it has become double, 10 centimeters square, that is A1. So this is absolute growth. In a particular time period, the 5 centimeters, the leaf size has expanded, it has become double, 5 centimeters square has become 10 centimeters square. When you come to this leaf, initially it is 50 centimeters square, later on it has become 55 centimeters square. So B, and later on B1 is value is 55 centimeters. At a particular time period, it has increased 5 centimeters square. And if you compare these two, in this leaf has shown more relative growth rate, where it in a particular time period has become double, but it has not become. So which is called the relative growth rate. When you compare the growth rate of two organs, like if you take a leaf, in this leaf has become double in size, in the area expansion. It has become only 50 has become into 55 centimeters square, which is a relative growth rate. So here if we take relative growth rate equation, that is the growth rate per unit time divided by initial size multiplied with 100. So in this picture there are two leaves, A and B. Okay. If we take the A leaf, growth in A leaf, initially it is 5 centimeters square, later on it becomes 10 centimeters square. So initial size is 5 centimeters square. Growth per unit time if we take, subtracting the final growth rate with initial, we will get 5 centimeters square. Relative growth rate, that is if it come to 5 by 5 into 100, it will be 100%. That is, the leaf has become doubled in leaf expansion. But when you come to B part, if you take B part, when you take the relative growth, initial size is 50 centimeters square. Final size is 55 centimeters square. That is, if we want to calculate growth rate per unit time, it is 55 subtracted with 50. Then you will get 5 centimeters square. Initial size is 50 centimeters square. So 5 divided by 50 into 100 will give only 10%. So leaf A if you take, it is 100%, and leaf B if you take, it is of only 10%. So in this, if you take in these two leaves, which leaves has shown more relative growth means it is this leaf A. Okay. And leaf B has shown less than when compared with the A. So this is how the relative growth rate is compared. So leaf A, which is of 100%, the relative growth rate is 100%. Whereas when you come to this B, it is only 10% of its initial size. So this is how we can calculate a relative growth rate. This is how we calculate relative growth. Okay. In growth, that is in the process of growth, we discussed that there are different stages like cell division, cell elongation, cell differentiation. In differentiation, we know that they will attain a specific function. They will attain a specific function, which is called maturation, which leads to maturation. Okay. So basically, when you come to meristems, which are young tissues of the plants, where they show active cell division. So these meristems will divide and form new cells. They will be in the process of continuous cell division, forming new cells. After a few days later, after some time, some of the cells will undergo differentiation. That means they will attain a function. So here differentiation means during this process of differentiation, the cells will lose the property of cell division. They lose the property of cell. Once they undergo differentiation, they cannot divide. They lose that capacity. Okay. So that is they will become mature cells. So they will become mature cells, which are called That is, the process is differentiation. So these mature cells, okay, which has lost the property of the cell division, okay, it has lost the property of cell division. Again, they undergo de-differentiation. Okay. Again, they undergo de-differentiation. This is called when mature cells will, by a process de-differentiation, when they gain the property of cell division. So which is lost in this, again they will gain the property. So that is called de-differentiation. Okay. That is called de-differentiation. And these de-differentiated cells again will lose the property of the cell division. This is called redifferentiation. So this is called redifferentiation, which can be seen during secondary growth in the plants. ये तीन प्रोसेस हमें देख सकते हैं, प्राइमरी ग्रोथ और सेकेंडरी ग्रोथ. During primary growth, some meristems on differentiation will form into mature cells. Okay. For example, if we take stem, in dicot stem, interfascicular cambium is formed from parenchyma cells, that is medullary rays. They are actually mature cells which are formed by differentiation, that is, they undergo de-differentiation. And again, from this interfascicular cambium, they will form different components, that is xylem and phloem, the mature tissues, which lose the property of cell division, which is called again the redifferentiation. So this is about the differentiation, de-differentiation, and redifferentiation, which can be seen in primary growth and secondary growth. You can see here, this is meristematic cell. On differentiation, it will form a permanent cell. That means they lose the capacity of cell division. So the capacity of cell division is lost. Again, on differentiation, they will gain the property of cell division. Okay. Just like formation of interfascicular cambium from parenchyma. When you look into the secondary growth in dicot stems, so during secondary growth of dicot stems, that is, the parenchyma cells in the medullary rays will form interfascicular cambium, which is a de-differentiation. Again, it will form permanent cells, different components of permanent cells, which by redifferentiation. And this interfascicular cambium, which is formed particularly during secondary growth of dicot stems, which can be seen in the secondary growth of dicot stems. So which is a process which occurs in dicot stems, which we can see secondary growth, during which cambium is formed, from cambium secondary xylem, secondary phloem is formed. The same we can see even in cortex also, which is called cortical division, that is in stellar growth and cortical growth. In cortex also from parenchyma cells cork cambium is formed, which is dedifferentiation. Cork cambium will form cork cells, that is the mature cells, okay, and which is a redifferentiation where cork cells will lose the property of cell division. You can take the best example for this also you can take is the cork cambium, which is a secondary meristem. These are called secondary meristems. Initially, it will be primary meristems. These primary meristems, when they lose the property of cell division, they form permanent cells. Again, they will, on dedifferentiation, they will form cork cambium, which is a secondary meristems. Again, by cell division, they will form permanent cells, which lose the property of cell division by redifferentiation. So this is about differentiation, dedifferentiation, or redifferentiation. You can look into this, the flow of this differentiation. Cells will divide, that means they will form and they will undergo elongation, maturation, and cells will be given a specific function. That is, once cells are given a specific function means they are mature cells or permanent cells, which is called cell maturation. And if you look into differences between dedifferentiation and redifferentiation. In dedifferentiation, the process by which they will attain, that is, they specialize because they lose their specialization and again become simplified and generalized. That means again they will gain the property of cell. Redifferentiation, that is, again they come into specialized form, that means they lose the function of cell division. They act as a meristematic tissue like interfascicular cambium, cork cambium, un meristem. These all are secondary meristems which are formed by dedifferentiation. And again in redifferentiation they will form specialized tissue. So these cork cambium, interfascicular cambium, wood meristems, these all will produce new cells at a particular part. But whereas when you come to the cells which are formed by redifferentiation, they will function at a specific, they will have a specific function. You can look into the example. Interfascicular cambium and cork cambium, which are fully differentiated, they are formed from parenchyma cells. And from this cambium, we can see formation of secondary xylem and secondary phloem, like tracheids, vessels, xylem fibers, xylem parenchyma. These all are the which lose the function of the cell division. The same thing if we take the phloem, sieve tube elements, companion cell, xylem is phloem fibers, phloem parenchyma. These all are the secondary, that is the permanent tissues. So this is the difference between dedifferentiation and redifferentiation. So till now अभी तक हमें पढ़ा है ग्रोथ में अर्थमेटिक ग्रोथ, ज्योमेट्रिक ग्रोथ, और इसमें हमें पढ़ा है ग्रोथ रेट, एब्सोल्यूट ग्रोथ रेट, रिलेटिव ग्रोथ रेट, डिफरेंशिएशन, डी-डिफरेंशिएशन और री-डिफरेंशिएशन. अभी हमें देखेंगे डेवलपमेंट. डेवलपमेंट बोले तो क्या है? डेवलपमेंट इज द sum of the growth and the different changes which occur in the life cycle of a plant. Development means it starts from germination to ends with senescence. Whatever the changes takes place in an organism in its life cycle. That is, if you take a plant, starting from germination to senescence, whatever the changes are taking place, these all will come under development. So growth is a part of development. So growth is a part of development. Here you can see in this picture, this is seed. Seed germination. Root system has formed, shoot system, later on flowers are formed, again they show reproduction. These all will come under development up to senescence. Up to senescence. Here you can see the sequence of development process in plant cells. It starts with meristematic cells, plasmatic growth. Again, some cells will form into meristematic cells. Elongation. Later on differentiation, they undergo maturation, they will form mature cells. These mature cells will undergo senescence, which leads to death. Senescence of leaves, senescence of roots. A पूरा final stage को क्या आता है कि senescence leads to death. Whatever it is, finally it reaches to death. These all will come under development, whether it is plasmatic growth or relative growth, whatever it is, these all will come under development. During this process of development, there are a lot of changes in the plant organoids. You can see different shapes, different sizes of this. Okay. Like if you take one term, plasticity, that is the ability of plant to follow different pathways, okay, depending upon the environment. For example, if you take cotton and coriander, the leaves at the younger stage are different from the maturation stage. Okay. You can see different shapes of the leaves, okay, depending upon the environment. Suppose when you take a leaf in its juvenile stage, that is at initial stages, if you take the leaf, and if you take the leaf in mature plant, there will be in different shapes. So this different forms of leaves, इसको क्या बोलते हैं कि heterophily. Hetero means different, phily means leaf. Okay. So if you look into this heterophily in larkspur, you can see different shape of the leaves. Heterophily in buttercup, which is a water habitat plant, where if we compare the shape of the leaf in a juvenile plant means the young plant and a mature plant, there will be of different shapes. इसको क्या बोलते हैं कि heterophily. इसमें हम देख सकते हैं environmental heterophily, developmental heterophily. Depending upon the life stages, there will be different forms of the leaf. इसको क्या बोलते हैं developmental heterophily. Depending upon the environment, some plants which live in water, पानी के अंदर रहने वाले लीफ शेप डिफरेंट रहता, पानी के बाहर रहने वाले लीफ शेप डिफरेंट रहता. इसको क्या बोलते हैं इकोलॉजिकल हेट्रोफिली. ऐसे during development you can see there is a lot of changes in parts of the plant, not only in leaf, even you can see, look into stem, root, like this. Okay, like you can see some adaptations in plants. Okay, like modifications in roots, modifications in stem, modifications in leaves, depending upon the life pathways and environmental conditions. For example, if we take open shell, this may leaves are modified into spines, which is a change. Okay. Like this, in some plants, leaves are modified into tendrils. Not only leaf, even buds, axillary buds, terminal buds are modified into tendrils. In some plants, they are modified into thorns. Like you can see, phylloclade, phyllodes. Like this, you can see many modifications in plants, which is also called plasticity. Okay. It depends on the life cycle and the environment in which they are living. Okay. There are different phases of life, like juvenile phase. Okay. Next is growth phase, and next is senescent phase. If you compare the structures in all these three phases, there will be different shapes and structures of a particular part, which is called plasticity. And when you come to leaf particularly, if you see different shapes of the leaves, which is called heterophyly. So which is called heterophyly. You can see heterophyly in cotton, coriander lines. Okay. So अभी तक हमें डिस्कस किया growth, development, and during development we discuss plasticity and heterophyly. इसमें इस सेशन में पूरा हमें डिस्कस किया कि ग्रोथ में अर्थमेटिक ग्रोथ, ज्योमेट्रिक ग्रोथ, रिलेटिव ग्रोथ, absolute growth, और डेवलपमेंट में मेरिस्टेमेटिक सेल्स, डिफरेंशिएशन, डी-डिफरेंशिएशन, री-डिफरेंशिएशन, प्लास्टिसिटी, हेटेरोफिली, पूरा हमें डिस्कस किया. इसमें वेरी इंपॉर्टेंट है कि the terms which you have come across, like arithmetic growth. What is the mathematical expression? LT is equal to L naught plus RT. Geometric growth. Okay. When you come to mathematics, W1 is W naught e to the power of RT, which is a mathematical expression where it shows sigmoid growth curve. Absolute growth or relative growth. How to calculate relative growth? Relative growth rate is growth rate in the initial size divided by final size into 100. You will get a growth rate. Okay. And you can compare the growth rate observed between two parts. Okay. And after this, if you look into this, next is the differentiation, de-differentiation, re-differentiation. डेवलपमेंट में क्या है, डेवलपमेंट में क्या होता है, व्हाट आर द चेंजेस, उसको क्या बोलते हैं कि प्लास्टिसिटी और हेटेरोफिली. सो दिस इज अबाउट द ग्रोथ एंड डेवलपमेंट. Follow us on social media, SPSVERSITY. Thank you all. We'll meet in the next session.